All right, guys, this is the second time I've had to do this one, so hopefully it'll be better. All right, so last video we left off discussing reception. Reception of a signal, oftentimes from the external environment. Sometimes it is from another cell, sometimes it's from a completely different organism. And we are on our pathway through, or on our march through this process known as the signal transduction pathway. So this video is going to cover the process of transduction, which you see in front of you. The picture in front of you shows the reception of the signal, the external signal, by one of a couple different types of protein receptors. Some of these protein receptors can be found embedded in the cell membrane, like the one you see in front of you right now. Others can actually be found floating inside the cytoplasm, which are called intracellular receptors, okay? And depending on the nature of the chemical signal that is being utilized to send a particular message, it may have to use one of those receptors or the other, okay? So if it's like a non-polar signal, it's going to enter the cell through the cell membrane, and it's going to bind with the receptor, intracellular receptor in the cytoplasm. If it happens to be polar or charged ion or something like that, it's going to have to utilize the receptor that's embedded in the surface. Now, from there we had got into specifics about these different specific receptors. Three of them we talked about: G protein-linked receptors, receptor tyrosine kinases, and ion channels. Those are three specific ones we talked about that have different ways of working and receiving the signal. Now, what we're going to get into next is the transduction of that signal, or the sending of that signal through the cytoplasm until it elicits some kind of cellular response. And we call this process transduction, okay? The movement of that signal from the outside of the cell all the way through the inside of the cell until it can actually do something, okay? And so this transduction, I like the title of these slides, is a cascade of molecular interactions and relay signals from receptors to target molecules in the cell. So that's what we're going to talk about. How do we get that signal from outside the cell, where it is kind of monitored, right, to the inside of the cell where it can actually do some good, okay? And transduction involves multiple steps, and you'll notice that right off the bat when we start to get into the specifics of transduction. And the reason that transduction involves multiple steps is because it can amplify the signal that started out as one molecule. It can amplify it into a signal that includes millions of molecules. And so that's one big perk of having these multi-step pathways, is that you can amplify a signal greatly. Okay, you can start with one molecule or a few molecules and produce a massive response as a result. Okay, as a result of using this. Multi-step pathways also provide opportunities for regulation and coordination. You're going to see an example of a cellular response in the final video of this that is very specific, and you're going to understand why you would want to be able to regulate the specific pathway that we're going to ultimately talk about, okay? And so I'm going to leave you hanging at that, because it's actually kind of an interesting pathway to talk about. But the minute you talk about it, you can be like, yeah, we probably should regulate that pathway. That'd be a good idea. You know, if you used a simpler transduction pathway that involved maybe just one step, right? That would be the easiest for the cell, wouldn't it? And it would be the least energy intensive, but it would also be the least flexible, the least variable. It would be the one that you can't change at all. Okay, it's just like, oh, here's one thing, and it goes and tells this. You couldn't do anything, and it might be really, really slow on top of it. Okay, so during this section here, we're going to spend ample time talking about what this transduction pathway is and how it works, as well as the molecules involved. Okay, so to get us going here, I want to talk briefly about second messengers. There are second messengers which basically carry the first message to the first transduction pathway. These are not technically part of the transduction pathway as we're going to talk about in specifics, but these second messengers are important nonetheless, okay? So that very first ligand signal, the chemical signal that is seen on the outside of the cell and interacts with one of our receptors, we can call that the first messenger, okay? And these second messengers basically take the interaction of that first message and the receptor, and they relay that message to the pathway, okay? And oftentimes these are small, non-protein, water-soluble molecules or ions, so they're primarily found inside of the cell. They don't diffuse into the cell. They're kind of just found right underneath the cell membrane. And the second messenger participates in these pathways that are initiated by some of the specific proteins that we talked about, like G protein-linked receptor and receptor tyrosine kinase, okay? Some examples of these things are listed below here. All right, right down here, cyclic AMP. Let me go ahead and get the pointer out. Cyclic AMP is a specific second messenger. Calcium ions can be considered second messengers, and inositol triphosphate, which is known as IP3. All three of these things are considered second messengers, okay? And I'm going to show you a picture of a couple of them here. The one you see in front of you is cyclic AMP, okay? Now, when we talked about G protein-linked receptors, this top portion of the picture should look familiar to you, okay? We talked about how the G protein-linked receptor binds the ligand, and once it does that, it is activated, right? And then it activates G protein, and then G protein goes and activates something else, right? That's another thing I want to remind you of, is that when we are talking about these molecules, we're talking about sequential activations, and these second messengers help with these sequential activations. Okay, and we can tell that these activations have occurred based on two things provided in our text and in the pictures. Number one, a shape change. The molecules that have been activated change shape. Okay, and number two, in the pictures that we see, the molecules that have been activated actually have this kind of glowing yellow aura around them, okay? Now, one thing to also remind you of is that molecules that are activated can then go and activate other molecules that are inactive, okay? All of these things are going to become vitally important when we start to discuss how that signal transduction pathway works. And all of these things that we see up here get activated or can activate other things, okay? So now this final protein in the string that we see here is now given a name. It's called adenylyl cyclase. You don't have to worry about the memorization of that particular fact. But adenylyl cyclase is going to then effectively activate this second messenger called cyclic AMP. Cyclic AMP is then going to activate our signal transduction pathway. That's what this molecule here represents, and we'll talk more about that in a second. And so what you can see is that these second messengers are kind of like the go-betweens or the relay molecules that connect this receptor complex up here to the pathway down here. All right, so the second messenger is kind of like this relay molecule. It's seen even better here with IP3 and calcium. Okay, IP3, here we see the G protein-linked receptor complex. Okay, all this stuff is part of the G protein-linked receptor complex. And we have IP3 down here, which acts as kind of like the go-between for our pathway. Okay, the pathway is right here. So, you know, these two diagrams actually show quite a bit of detail, especially the one you're looking at right now. I'm not going to require you to know that much detail about what second messengers do, but I want you to know their role in the entire process known as signal transduction, okay? I want you to know that they basically take the message from the receptor and they relay it to the signal molecules, okay? So do keep that in perspective. Remember to yourself, okay, we don't need to know specifics, but we need to know that IP3, calcium, and cyclic AMP relay this message from the receptor to the signal molecules, okay? I think that's of most importance. So let's talk specifically about what the signal transduction pathway is and what it does and kind of how it works, okay? This statement right here describes to you how the signal transduction pathway works. The signal transduction pathway is effectively a sequence of many proteins that all activate one another. Okay, so like falling dominoes, a receptor protein, the very beginning part of this, right, activates another protein, which activates another protein, and another one, and that one activates another one, and then that one activates another one, until the final protein that produces some kind of cell response is activated. Eventually there is a stopping point to this, but oftentimes there are 3, 4, 5, 6, 7 steps in a signal transduction pathway. And each one of those steps is represented by a particular protein that is activated. And then it activates another one. And then it activates another one. Okay? So this idea of sequential activation that I've already discussed becomes vitally important to understand, right? So at each step, the signal is transduced into a different form. And usually this form is in the form of a shape change. Okay? And so when we see some of the diagrams coming up here, I want you to be aware of this, that every time you notice a new protein has been activated in the pathway, that means that the protein has changed shape, and it has picked up a little glowing yellow aura. If you guys remember how important those things are, right? Okay, so how do these proteins become activated? All right, I've used this kind of amorphous term, this term activation, and you may be wondering, well, how does that activation occur? Well, you're going to get part of that story right now, okay? Let me go ahead and make it so I can draw on the slide here, because I've got something that is on the slide that we haven't really talked about yet, and that's a function of taking these chapters out of order. We will not learn about this term phosphorylation until chapter 8, which is until next unit, but we're doing chapter 11, so we should have learned about it already. Okay, yeah, it's my fault a little bit, but I'm going to give you a crash course in phosphorylations, okay? In fact, in the previous video, I believe I've already kind of alluded to it, though I didn't use this term phosphorylation, okay? But this idea of phosphorylation is what activates the sequential proteins, okay? So let's talk about it. ATP, right? We all know what ATP is, adenosine triphosphate, and I'm going to draw a little it's a crummy drawing of it, but it'll help me get the point across what I want to do. Okay. So ATP stands for adenosine triphosphate, okay? Now, one of the things that we will eventually learn in chapter 8 is that if we want to harness the energy, because ATP has energy associated with it, if we want to harness the energy of ATP, what we actually have to do is chop off, let me change the color here. What we have to do is actually chop off one of the terminal phosphates, okay? And that would be this guy right here, would chop it off. And when we do that, what we do is we effectively release the energy. We can harness the energy. We can make it do something for us. And in this case, when we talk about the sequential activation of proteins, we can use this energy to activate a protein. Okay, so let me go ahead and draw a protein. I'm going to draw it in purple because a lot of the proteins we see in our book are shown in purple. Okay, so that is a protein, and we want to activate it. And so when we activate a protein by phosphorylation, we use ATP, we chop off the phosphate group. That phosphate group actually gets attached or added to the protein, okay? And at this point, this protein has been activated, okay? It has been phosphorylated. So by taking one of these terminal phosphates off of the ATP and attaching it to a molecule, we activate it, okay? Effectively, we impart the energy of the ATP to this protein right here, okay? And so we now have a active phosphorylated protein, okay? And now it can go activate other stuff. All right, let me go ahead and advance this slide a little bit, okay? This phosphorylation needs help. The process of phosphorylation needs help, and it gets its help through an enzyme called protein kinase, okay? The enzyme called protein kinase is vitally important. Let me go ahead and erase all ink here. And I should just get rid of that. And I'm going to advance the slide here one more. It gets its help from protein kinase. Now, in this process of protein activation, protein kinase is kind of like an on switch, okay? And I'm going to go ahead and use green. Okay? Phosphorylation is like an on switch, okay? And it is done by protein kinase. Now, opposite of that would be something called dephosphorylation, and dephosphorylation is like an off switch. And the enzyme known as phosphatase reverses phosphorylation. And effectively what you see with dephosphorylation is you see the removal of that phosphate group. So if we had a protein that had been, there's our protein, that had been phosphorylated, like that, dephosphorylation would cause this phosphate to fall off, okay? And the enzyme that is responsible for this process right here occurring is called phosphatase, okay? So those are some facts that you're going to want to make sure you remember, okay? So, protein kinase acts as the on switch, and it phosphorylates a protein. Phosphatase enzymes act to dephosphorylate something or take the phosphate group off. Okay? Now, this phosphorylation-dephosphorylation system acts as this molecular switch, and it turns certain activities on or off. Or it can turn certain pathways on or off. Okay? Now, you can disregard the bottom thing here. If you really want to know about how this relates to a stupid 80s movie called Over the Top, I would be glad to talk to you about it in class, but you're going to have to remind me. It's one of the stupidest things over here. So if you want to hear it, you're going to have to remind me. I'm not going to waste any more time on it today. Okay? But if that link works, go for it. I doubt it will. All right, so how does all of this stuff come together? How does this idea of sequential protein activations and second messengers and phosphorylations and dephosphorylations, how does it all come together into the relaying of the signal through the cytoplasm? Because as at the beginning of this entire process, talking about all this stuff, I said that's what we're going to talk about, is taking this signal that was received at the cell membrane through the receptor and talking about how it gets transferred through the cytoplasm to ultimately elicit some kind of cell response. That's what we're going to talk about here in this particular picture. Okay, this particular picture is kind of like the capstone. It's the one that you finally get some of the answers and finally get all the stuff put together in one singular spot. Okay, so let's go ahead and just to talk about it briefly. Let's talk about it from the very beginning. Let's talk about the reception of the signal molecule, which we see up here. The ligand has let's use this bright green. The ligand has, I don't like that at all. We'll go back to red. Has bound up to the receptor molecule. Okay, so we've received the signal from the external environment, and the receptor or the receptor molecule is going to then relay that signal into the cell. Okay, and we know that this receptor has been activated because we see it's got a little yellow glowing aura around it, right? And we know that if it's activated, it can then activate something else. Okay, now this right here is our second messenger. Okay, that is our activated relay molecule. I said, okay, it takes that signal from the receptor molecule embedded in the cell membrane, and then it brings that signal to the pathway, okay, to the transduction pathway. That's what we're going to talk about now. Okay, so let me erase some of this stuff so we can see a little bit better. All right. So at this point we have an activated relay molecule, right? And we want to then activate a bunch of other relay molecules, or a bunch of other molecules in the cytoplasm that will relay this signal through the cytoplasm and eventually elicit some cell response. But to do that, we have to activate some stuff, right? Because activated things can then send their message to the next molecule, okay? And that's what we see here. We want to activate this molecule right here. It says inactive protein kinase 1, okay? And we want to make it active. And so all we have to do is bring it in close proximity to this other molecule right here that is already activated, okay? And what we see happening is that it has been activated. I'm going to circle it right here. Protein kinase 1 has been activated. All right. So how do we know it's been activated? We look at the shape, right? Original protein kinase 1 has kind of this oblong oval shape, right? And activated protein kinase 1 has just a sphere shape. Okay, great. Now, so we've got our first protein in the signal transduction pathway activated. It is called protein kinase 1, all right? We have to relay that message to the next step in the pathway, okay? And so to do that, we have to bring inactive protein kinase 2 into close proximity with this activated protein kinase 1, okay? Now, what you'll notice here is that we've now incorporated ATP, right? And we're going to start to phosphorylate all of these sequential proteins, okay? The reason that protein kinase 1 did not become phosphorylated was because this relay molecule was already activated, okay? And these molecules, these protein kinases, need to have, these sequential ones need to have phosphates to activate them, okay? So, we take protein kinase 2, we add some ATP, and we phosphorylate it. That's what we see has happened right here. We've added that phosphate group to protein kinase 2. And so we see that it has changed shape. It is no longer an oval, it is now a sphere. And so protein kinase 2 is active. Okay? Check that one off. Next, we want to activate protein kinase 3. Okay, so we're going to bring this protein kinase 3 into close proximity with protein kinase 2, which is already activated. We're going to incorporate some ATP, and we're going to phosphorylate active protein kinase 3 and activate it. Okay? Again, we see the shape change, and we see the glowing yellow aura. Finally, we're going to have this last protein in the sequence be activated. It's a different protein. It's the final protein, but we still have to use the same process. We bring it in close proximity to protein kinase 3, which is already activated. It will phosphorylate this final protein, and now it becomes active as well. Once this final protein is activated, it will elicit some kind of cellular response, finally, right? And so that's what we're going to eventually talk about in the final video. Now, let's go back right up here and kind of put a bow on this picture, okay? We've got to make it look a little bit prettier. I'm going to use a different color just so it sticks out a little bit. I'm going to use this pink one. Yeah, why not? This backwards arrow that we see right here, okay? We see this molecule PP, okay? PP stands for phosphatase, okay? And what that does is it shuts the system off. Once this has been activated and done activated protein kinase 3, we're going to shut it off. We don't need it anymore. And so phosphatase basically chops that guy off right there, that phosphate, and that's what you see falling away over here. Okay? And it reverts, protein kinase 2 is going to revert back to its inactive form. Okay? And then we see the same thing here. Phosphatase will chop off this phosphate, which we see float away over here, and protein kinase 3 reverts back to its inactive form. So I know that there's a lot going on here, guys. I really do. But I've got a few animations that I think will allow you to see what's going on a little bit more a little bit more streamlined. And when you see these animations, I think it really does put everything into perspective, and it allows you a better understanding of how this stuff actually works. Okay? So I fully expect there to be a lot of questions when we get done with this, and I want to make sure that you guys are writing things down as you read all of this and as you watch these animations. Okay? So I'm going to go ahead and start the animations, and then I'm going to kind of be quiet while this guy goes through them.
Receptors that are plasma membrane proteins usually affect the cell through multi-step signal transduction pathways. These pathways allow for amplification of signals and signal coordination and regulation. Signal transduction often involves smaller molecules called second messengers and protein phosphorylation by enzymes called protein kinases.
Okay, so the first couple of these animations go into second messengers and their role in this entire process. So kind of keep that in perspective, but I want you to watch all of them because it shows you the moving parts and how they interact.
Second messengers are small, non-protein molecules that act as intermediaries in signal transmission. Two important second messengers are cyclic AMP and calcium ions.
Cyclic AMP acts as a second messenger in pathways initiated by both G-protein-linked receptors and receptor tyrosine kinases. Click on the signal molecule, the first messenger, to see how cyclic AMP acts in signal transduction.
The hormone epinephrine acts via cyclic AMP as a second messenger. An epinephrine molecule docks with a receptor protein, which acts through a G protein to activate the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP, or CAMP, which quickly diffuses through the cell and triggers further steps in the signaling pathway. In this cell, we show it activating a protein kinase. Calcium ions also act as second messengers in signal transduction pathways. Click on the signal molecule, the first messenger, to see how calcium ions...
This time we will show a receptor tyrosine kinase, but G-protein-linked receptors can also initiate release of calcium ions. The signal molecule docks with the receptor and activates an enzyme called phospholipase C. The enzyme cleaves a small molecule called inositol trisphosphate, IP3, from a certain kind of membrane phospholipid. IP3 docks with a calcium channel on the endoplasmic reticulum. The channel opens, releasing calcium ions into the cytoplasm, where they activate proteins that carry out cell responses.
Okay, so I just want to kind of put this into perspective. The stuff that you just saw is pretty high level, the intricate workings of second messengers and how they move from place to place and interact with other things and turn other things on. We don't need to know that much detail about it, okay? I wanted you to see what those second messengers do. I don't necessarily need you to memorize every single step of what they do. I just want you to see their role in all of this, okay? The fact that they basically take this received message and they relay it to the signal transduction pathway, okay? In fact, I used, to put this a little bit easier, good analogy, I used an analogy about falling dominoes, right? That each one of these, I'll come back to this in a minute. Each one of these proteins that's activated, okay, is like a domino that's being pushed and falls into the next one, right? So you can think of the second messengers as the finger that pushes the first domino, okay? So this would be like the first domino, protein kinase 1. It's that second messenger pushes it and the rest just fall down, okay? That's how I would like you to kind of characterize the second messengers and the role that you have to understand with them.
Enzymes called protein kinases are important links in many signal transduction pathways. Click on the signal molecule to activate a signal transduction pathway involving protein kinase. A protein kinase catalyzes the transfer of phosphate groups from ATP to another protein. More often than not, the activated protein is also a protein kinase, which may act on still another protein kinase. One kinase may activate many molecules of the next type of kinase in the chain, thus amplifying the signal until the last kinase activates many molecules of the protein that carries out the final cellular response.
All right, so I really do want you to see these animations. So if for whatever reason they didn't work or it didn't translate very well or you couldn't hear the guy, make sure you let me know so I can show them in class, okay? And we show them live or whatever. Because I think they are really going to be beneficial to you understanding how these things work. And you can see them in motion in your mind, but you can take this kind of complex diagram and put it in motion. That really does, I think, help make sense of it all, okay? Now, as I said before, the next video is going to be getting into specifics about this cellular response. So I've talked about this ambiguous cellular response. What is it? Why won't he tell us more? Well, I'm going to give you some detail about that, and we'll talk about a very specific cellular response that's actually quite interesting. That is kind of the final countdown, the final step of signal transduction pathways, where we can talk about what it means to receive this signal, transmit it through the cytoplasm, through transduction, and then get this really cool cellular response at the end. Okay? So that'll be in the fourth and final video, and I'll see you guys soon.